So it's my pleasure to introduce Dr. Caitlin Price. Before we start, Caitlin performed her master's degree at the University of Florida, Department of Soil and Water Science, and his topic of her master was on carbon mangrove. Then from the same university, his PhD, her PhD on permafrost carbon. And then finally, her postdoc was in Berkeley Lawrence National Lab on world soil carbon warming. Since 2017, Caitlin is a professor of biology at Dartmouth College. So the presentation of Caitlin will highlight the response of deep soil horizon to climate change, a very timely and significant topic, particularly in the context of carbon neutrality for climate action. And the presentation is divided in two parts. Part one will stress the vulnerability of soil carbon to climate change and when does the deep soil not respond. And the second part is more on global driver of soil carbon persistence. So I hope you will enjoy the presentation, and Caitlin, I'm sure, will be very pleased to respond to any question. Thank you.
So yes, I am talking to you all from France, but still on the internet. So maybe one day I'll get to come back here and meet all of you in a post-COVID world. But in some ways, this is good because we'll probably have some of you from all over the world that are able to watch this because we are on the internet. So today I'm going to talk about deep soils and climate change, from experiments to meta-analysis. So just some brief background on soils to start if you're not a soils person. And so soils provide essential ecosystem services. I put up this picture that I saw in the Musée d'Orsay this past weekend, which I loved because it's just a beautiful depiction of soils. And soils, they retain water, they provide nutrients to plants, and they perform a really important ecosystem service of storing carbon. So that ecosystem service is the one we're going to focus on today. And soils store about 3 times as much carbon as is currently in our atmosphere. So currently the best estimates of soil carbon are that there's about 3,000 petagrams. That's 3,000 times 10 to the 15th grams of carbon stored in soils globally. And so the atmosphere holds 800 petagrams of carbon. So the big million-dollar question when it comes to soils and climate change is how much of this carbon will be released to the atmosphere as a result of a changing climate, a both warmer climate and a climate where precipitation might be drier, wetter, or just vastly different than we have experienced in the past. And currently we have a lot of models that can predict terrestrial feedbacks to climate change. And these models are all highly uncertain. This uncertainty, this is from a paper in 2014. With the new models, I should update this because the uncertainty is even greater in the next generation of climate models. So in this paper, it looked at 11 models and found that there was an over 600 petagram difference in terms of the amount of carbon that the land would either store or release as a result of climate change until 2100. So this is equivalent to 1,200 ppm CO2 in the atmosphere and 3 to 6 degrees of warming. So this is a massive degree of uncertainty. And much of this uncertainty, not all, but much, I would say the majority of it, comes from our lack of understanding of what's going to happen to soil carbon. So globally, half of soil organic carbon is actually stored below 20 centimeters. However, traditionally, agronomists and soil scientists have really focused on the top 20 centimeters, top 30 centimeters of the soil profile. And so we have warming studies that have looked at the response of this topsoil carbon. They focus on it because that's where the majority of the roots are with plants. And honestly, it's a lot easier to study the surface of the soil than it is to study deep soil, especially when we're looking at deep soil responses to change. And so because of the fact that it's much harder to do these studies and that people traditionally did not think of it as important to understand what was going on in the deep soil, we are really uncertain about the response of soil carbon at depth to climate change. And one question I always get asked is, well, maybe the deep soil won't warm. maybe it will not be affected by climate change. How do you know that the deep soil will warm just like the surface soil does? And so this paper used the CMIT model prediction, so similar models to the last graph that I showed. And these models predict, and we have a much better understanding of these temperature dynamics than we do the carbon dynamics in the models, I will say. So these models all showed that the surface and deep soil, so soil basically in the top 10 centimeters and at 1 meter depth, they warmed with the air and basically in lockstep with each other. So when the surface soils are warming, the deeper soils are warming too. And they warm at pretty much the same rate as the air. So predicted increase of 4.5 degrees by 2100 based on scenario 8.5. And that there's no difference, again, there's no difference between surface and deep soils with this warming. You might see that there's a bit of decoupling between soils in the atmosphere toward the end of the century in the models, and that has to do with snow dynamics in the Arctic. So we know deep soils are important, and we know that soil depth is really important. All soil scientists, like one of the first things they do when they go out to a site is they dig a deep pit in the earth, and they describe the soil by horizons. And you can see in this agricultural soil on this mowed field that had been plowed for a long time, we have this very large 30-centimeter plow layer. And then you can go down the soil profile, you see it gets more light in color, there's less carbon. There's a lot of carbon here in the plow layer, there's less. As you go down a soil profile, you can see changes in the amount of carbon, changes in the mineralogy, in the texture, and how it can hold water. So we've always known that depth matters when it comes to soils, but yet we haven't been looking at the dynamics of how different soil depths are responding to climate change. And we know that in terms of the mechanisms controlling soil carbon, that those change a lot with depth. We know this because the age of carbon increases as you go down in the soil profile. So it seems that the carbon is more persistent and older as you go down the soil profile, and maybe that might make it less likely to respond to climate change. But again, we're just starting to test this. So what changes with depth? We have the biotic influence. It's high at the top. At the top, that's where most of the roots are. So we have a lot of rhizosphere, which is soil that's affected by roots and root exudates. There's high levels of organic carbon and high populations of soil microbes, both bacteria and fungi. As you go down in the soil, you have less and less roots and less of these really labile root exudates that the microbes love to eat. You have less organic carbon, and so you have less microbes. They're able to decompose the carbon as you go down with depth. Often, as you go down a soil profile, the mineral influence increases. And so sometimes the amount of reactive minerals that can actually absorb to carbon and protect it from being decomposed, those increases. And the amount of carbon associated with minerals versus free in the soil, which I'll talk about in a second, that increases as well with depth. There's big differences in temporal variability. And so at the surface, you have daily changes in temperature, seasonal changes in temperature, all of which are dampened deeper in the soil profile. Same with moisture availability. It can be, dry out a lot more at the surface and get really wet at the surface. And then as it percolates down the soil profile, those can be dampened, though depending on the soil, it might actually be much wetter at depth if you have a water table. The spatial connectivity between the organic carbon that microbes eat and the microbes themselves differs. So in the surface where you have a lot of rhizosphere, there's just a lot of organic carbon, a lot of microbes and roots, and they're all in the same place. And then as you go down with depth, you get these sort of hot spots of organic carbon and microbial activity. But oftentimes you might have a difference between where the microbes are and then where the organic carbon is, because the microbes will decompose the organic carbon, it won't be there. And then maybe there's a new root that comes down or a preferential flow path bringing organic carbon down. And that might be separate from where the high microbial population is. So it's just more spotty, and there's less connectivity between the organic matter and the microbes. So all these things change with soil depth, which leads me to believe that we shouldn't expect deep soils to respond similarly as surface soils due to warming and climate change. It's also really important that we start to understand these processes with depth, because the next round of soil carbon models are explicitly including depth. This includes the E3SM model coming out of the US Department of Energy. Even CLM now has different soil depths that are being considered. And so in order to verify these models and help parameterize these models, we should know how these processes that the models are representing differ across depths. So we know that soil carbon is heterogeneous. It's not all the same, and I alluded to this in the soil depth slide. In a very basic way, we can split it into 3 different pools of soil carbon. The first would be particulate. You can see here from these microscopy images that the particulate organic matter looks like plant parts. It's, pieces of wood, roots, leaf litter. Oftentimes it's identifiable plant parts that have not undergone a lot of decomposition. Some of this particulate organic matter can be stabilized within soil aggregates. So within a soil aggregate, you can have organic matter in small micropores, and that makes it inaccessible to microbes, and so it will be slower to decompose. Lastly, you can have organic carbon that is associated on the surfaces of soil minerals, and it is thought that much of this carbon is inaccessible to microbes and won't undergo the same amount of sort of enzymatic decomposition and attack that the particulate organic matter can. So the soil carbon is heterogeneous, and how accessible the organic matter is to being decomposed by microbes and then likely being responsive to climate change depends on which pool, whether it's particulate, aggregated, or mineral associated. So in the lab, however, we have to split it into these pools. And so soil fractionation is an attempt to split carbon into these different pools. And in models, they do this by splitting carbon into slow and fast pools. Models might have just one of each or a whole continuum of slow to fast pools. In the lab, we can do this through operational definitions by sieving the soil. So you could have pools based on the size of the particles, by soil incubations where the microbes fractionate it for you. So that fast decomposing pool is the amount of carbon that gets decomposed quickly by the microbes. And lastly, by density, which is what we'll be focusing on today. So with density fractionation, you can split the soil into a free light fraction, which is that particulate organic matter. This is what floats on top of your dense solution. It tends to have a low resonance time, so it only stays in the soil for years to decades. In tropical systems, that might actually be months. It contains fragmented root litter and fungal biomass, think hyphae, and is made up of relatively complex molecules. Next, you have the occluded fraction. This is what floats after you apply energy. So after you shake this dense solution in the soil together or apply sound energy. So it's thought to break up aggregates, and it tends to have an intermediate residence time between the free light and a heavy fraction. And then lastly, we have the mineral fraction. This is what sinks to the bottom. It's heaviest, and it is the carbon that is associated with soil minerals, oftentimes thought to be derived from leachate and microbial biomass, and tends to be of a simpler molecular structure, such as proteins and sugars. So it's thought that oftentimes this carbon has undergone microbial decomposition before becoming associated with organic matter, with soil minerals. And it tends to have longer residence times, up to centuries. So lastly, I just want to introduce you to the idea of radiocarbon ages. So these soil fractions that I just spoke about, I said they had different residence times. This time is based on radiocarbon ages, which can be thought of as our clock for the carbon cycle. So we can age carbon over hundreds to tens of thousands of years based on the radiocarbon that's produced in the atmosphere that then undergoes radioactive decay as it sits in plants and then in the soil. And so you see here the C14 depletion, delta 14C. It gets more negative the older the carbon is, and it can date back carbon to about 50,000 years. Next, we also have bomb radiocarbon, which can age carbon on a much shorter time scale of years to decades. And this is due to atmospheric nuclear weapons testing, which put a bunch of radiocarbon into the atmosphere in 1963. And so based on the amount of carbon that is of this bomb carbon in the soil, we can age carbon for about the past 60 years. So you see these delta 14 seed values are very, very positive, unlike these ones, which are very negative. And so the most positive value would be carbon from the 1960s, and it's been declining pretty fast as it gets diluted by fossil fuel carbon, which has no radiocarbon in our atmosphere. All right. So the big questions that I want to answer today are, how does depth affect the vulnerability of soil carbon to climate change, and which fractions of carbon are responding to climate change? I will first talk about warming responses from in situ experiments in forests and grasslands, and then briefly talk about a meta-analysis that looked at global drivers of soil carbon persistence, which I think can help inform the results of the warming experiments. So my overall hypothesis is that deeper soils will be less responsive to warming due to the increased protection of organic carbon by mineral associations. So I'm thinking that organic carbon in that dense fraction will not be responsive to climate change. The first study I'll talk about is from Blodgett Forest in California. This is a coniferous forest in a Mediterranean climate where it has warm, hot summers and cool, wet winters, oftentimes with snow. So this was the first warming experiment of a whole soil profile in a mineral soil. And so for this experiment, we had 3 pairs of control and heated plots. In the heated plots, we have 22 2.4 meter long rods that go into the ground surrounding this 3 meter diameter plot. And inside that are heating cables. So we heat the soil basically from all sides. We also have heating cables under the organic horizon and the surface of the plots to help warm the soil sort of from the top, like a traditional warming experiment does. So what's different than a traditional warming experiment, however, is that through this design, we can get more even heating throughout the entire soil profile. So our goal was to have it be +4 degrees. And you can see that from about 15 centimeters down to 100 centimeters depth, we have about +4 degrees of warming. We have less warming at the surface because we are losing heat from the soil surface. So the first results from this experiment showed that warming the whole soil profile resulted in a 34% increase in soil respiration with warming. This was not surprising and was in line with the increases that we saw in other forest studies where they did surface warming, though it was a bit larger of an increase than had been seen in previous studies. But what we really wanted to do was to see how these different depths were responding to warming. And to do that, we had to measure vertically resolved CO2 production. So we did this by inserting gas wells. Think of these as metal straws into the soil at 5 different depths. So we had these metal straws that are permanently installed within our plots at 15, 30, 50, 70, and 90 centimeters. We measured CO2 concentrations in these wells monthly, and then we were able to model production of CO2 at each one of these depths using Fick's law of diffusion, shown here. I can get more into that later if you would like. So this shows the CO2 production from the different depths in the soil profile. Red is warmed, blue is the control. And at all depths, we have increased soil CO2 production in the warmed than in the control. The 2 bottom depths, which have much lower rates of CO2 production, not unexpected given that they have much smaller amounts of organic carbon, are shown in this inset, but you can still see here that the 90 to 70 centimeter depth interval had significantly more CO2 being produced in the warmed plots than in the control plots. So overall, 40% of our response was from soils that were greater than 15 centimeters deep in this study. We also measured the Q10, which is a measure of temperature sensitivity. So the Q10 is the amount by which respiration will increase for a 10-degree increase in temperature. It's like a multiplicative factor. So if you have a Q10 of 2, a 10-degree increase in temperature will increase the respiration times 2. So it will double it. So what we found when we looked at the Q10 of all the different depths was that there was no difference in Q10 among depth. The average Q10 was 2.5. And so we found that deep soils were just as responsive to warming as surface soils. This was surprising and went against our hypothesis. So to look into why this was happening, I used radiocarbon values of CO2 throughout the profile to see if there are any differences in the age of the carbon being respired at depth or with warming. We found that the radiocarbon values throughout the profile were young. So 40 per mil here is just above, so just a little bit older than the atmospheric value at the time, which was 28 per mil. And there was no difference among treatments in the age of carbon. So what was the source of the carbon fueling this response to warming? Here are the radiocarbon values of the bulk soil in gray. So you can see that they get more negative, more depleted, and thus older with depth. The brown shows the radiocarbon values of the dense fraction, which also become older with depth. And then in black, we see the radiocarbon values of that free particulate fraction that's unprotected. And that fraction generally remains young with depth. And so I hypothesized that the free particulate fraction and its decomposition was what was driving the response to warming. And now we have later data that can show whether I was right or not. So first, was the initial response sustained? So the data I showed was over 2.5 years. We now have data over 5 years. So these dots here, black is warmed, white is control, and this is the mean flux of carbon. This is from the soil surface. And over time, we still have a significant warming effect where CO2 production is warmed, it's greater in the warm soils. It has gone down, so this effect has decreased over time, but not yet significantly. And the Q10 has also stayed high throughout these 5 years. So far, the warming response has been sustained. And what we found was that after 4.5 years, we saw a significant decrease in the soil carbon at depth. So we've been able to look at a change and see we've had an almost 40% decrease in the carbon stock at depth as a result of warming. And we were able to fractionate the soils again to see where this was coming from. So these are the amount of carbon in the particulate, the aggregated, and the mineral associated fractions, fractionated by density. And you can see here in the particulate fraction, that free particulate fraction that's not protected, we see a significant decrease in the warmed plots relative to the control plots. There's no significant differences at depth in the aggregated or mineral associated carbon pools. So it seems that my hypothesis was correct, was that this response, particularly the response at depth, was being driven by loss of this free particulate fraction that is not protected by soil minerals. So the takeaway from this experiment was that deeper soils are responsive to climate change. But what about other soils? So I have now moved to New Hampshire since then, and I have looked at other warming experiments. This one is an ongoing warming experiment in Hubbard, Brook, New Hampshire. So a very different climate and different forest type than Blodgett. Here we have relatively shallow soils. They're 40 to 50 centimeters deep because this is an area that was recently glaciated. It's a very cold climate with a mean temperature of 6 degrees Celsius. And instead of cambsols, these are on podzols. And these are hardwood deciduous forests and not a coniferous forest. So this experiment that I came in to make measurements at is called CCASE, the Climate Change Across Seasons experiment. Here we've had treatments for the past 7 years. This is a surface soil experiment, like many warming experiments are, but there's a little twist here in that there's a control, and then there are heated plots, which are heated to +5 degrees during the snow-free season only. And then there are another set of plots that are freeze-thaw cycle plots, which during the winter undergo at least 4 freeze-thaw cycles experimentally induced and are then warmed in the summer. So I sampled in 3 locations per plot. First, I found that buried cables at 10 centimeters, so the surface warming in this site was able to warm soils, deeper soils. This is temperature difference between control and the heated and freeze thaw at 10 centimeters and at 30 centimeter depths. These are plots of the CO2 production by depth. This time I'm showing not just an average, but how it looks over time. So we have the organic horizon, the 0 to 10 centimeters of mineral, and the 10 to 30 centimeters of mineral. And again, these are relatively shallow soils compared to those in my last experiment. We found that the mineral soil CO2 production did increase in the warmed plots. So generally we had greater CO2 production, that's in the red, than in the control plots, which are in blue, throughout the growing season. We also saw that the O horizon and the deeper mineral horizon decreased, the CO2 production decreased in response to the freeze-thaw cycles. So that's in orange. So what this shows is even though those freeze-thaw cycles are really only affecting and freezing and thawing the organic horizon, the 10 to 30 centimeters of mineral horizon does not freeze at all. Even though the top is being affected, we're still seeing a response in the deeper soils. Overall, the percentage of production that came from the O horizon was reduced by both warming and freeze-thaw cycles. And that in turn sort of made the carbon coming out of the mineral horizons more important and a larger proportion of what we're seeing in terms of the response to climate change. So again, we found deep soils were responding in a forest soil. So when does the deep soil not respond? Well, continuing our trip around the United States, there's an experiment that I've been collaborating on in Hawaii. So we were in Blodgett in California and then New Hampshire, and now we're at an experiment in Hawaii where we have a very warm temperature, 25.6 degrees Celsius, and very wet soils with 4,200 millimeters of rain a year. These are a special soil that are andisols, which means they have a lot of highly reactive minerals that can absorb onto organic carbon. And this is in a tropical rainforest that has been disturbed. So this is an experiment that I collaborated with Susan Crow and Casey McGrath of the University of Hawaii on. We're currently writing up the paper and we'll be submitting it soon. We call this soil warming on the cheap because instead of having these plots surrounded by 22 heaters each, we actually, across this landscape, put 5 deep soil heaters. You can see them in red on this map here. And the colors on this map just show what the CO2 surface production was like pre-treatment. So we use this random array of deep heating rods to create a range of temperature manipulations across our site. And in black, on this map, you can see where we had gas wells that were measuring CO2 production across the different depths. Overall, in this graph, we found no temperature response at depth. So this is the CO2 produced versus the change in soil temperature from the control. So again, we had a range of soil temperature manipulations at this site. In the 0 to 40 centimeter depths, we saw a significant increase in the CO2 produced as the temperature increased as well as an experimental temperature increase. So that was significant. We did not see a significant relationship in the 40 to 100 centimeter depths. So if surface soil responded, the deep soil did not. And we hypothesized that this was a result of the increased abundance of highly reactive minerals at depth. So this is poorly crystalline and non-crystalline minerals, iron and aluminum oxides here. And the amount of these increases with soil depth. So the surface is this light color, beige, and then blue and purple are the deeper soils. And you can see that the CO2 produced decreases as the amount of poorly crystalline and non-crystalline minerals increases in the soil. So here, deep soils did not respond, and they were likely highly protected by the minerals, so they were not accessible to microbes. We'll now look briefly to grasslands. So here, grasslands are less well studied in terms of deep soils, mainly because the way that people have warmed grasslands has always been by warming the air above the grassland, which it's unclear how whether that warms deeper soils or not. So I've been here in France for the past week to work with Abad Chabi on a plot in Lusignon, two experiments actually. Here we have a temperate grassland, and there are two sites where we have warming, a mowed grassland and a cropland. So here's the mowed grassland, and then here's the cropland currently planted with wheat. And so we were there sampling last week. So in each site, we have 3 pairs of heated and control plots. It uses the same design as my California project, where we have the 22 deep soil heaters around a 3-meter diameter plot. So this is very new, so I don't have any results to show you, unfortunately, but I can show you what I will expect to see based on an incubation I did of grassland soils in a mowed field in New Hampshire. So this was an incubation. So this is where the microbes kind of fractionate for you. And in this incubation, we incubated soils from a variety of different depths at both 7 and 17 degrees Celsius. Here we have the carbon loss per gram of carbon in the soil. And we can see that there's a significant temperature effect in the top 30 to 40 centimeters, where there's a lot more carbon that is respired at the warmer temperature than the colder temperature, but that there's no difference below 50 centimeters in the amount of carbon that's respired. So in this grassland, at least in New Hampshire, we did not see a deep soil response to warming in this incubation experiment, which again is not in situ. So we don't know yet what's happening in situ. Hopefully we'll have some results from Lusignon soon. So lastly, I want to briefly go over some global drivers of soil carbon persistence and relate those to what we learned in the warming experiment. So for this part, my question was, what is the relative influence of soil forming factors and depth on the abundance and persistence of soil carbon among different fractions? So by abundance, we are looking at the carbon concentration. And by persistence, we mean that radiocarbon value, that age of soil carbon. Again, we looked at those density fractions. So we had particulate, occluded, the aggregate fraction, and then the mineral associated fraction. And what do I mean by soil forming factors? This is the thing that you learn in intro soil science, where like soil is a function of CLORPT, which is climate, organisms, relief. So topography, elevation, aspect, that sort of thing, parent material, and time. This was a large-scale meta-analysis that looked at all these factors, but today I'm going to focus on 2, the results from climate and organisms and how they affect soil carbon abundance and persistence. So for this work, I used the ISRAD database. And this is a database that was developed by a lot of people worked on this. We had a Powell Center working group sponsored by the USGS in the United States. We had funding from Lawrence Berkeley National Lab. We had funding from the European Union. We had funding from the Max Planck Institute. So this was a real collaboration between scientists in the US and in Europe. And we worked on this for years to get a database of all the different soil radiocarbon data. So we then use this database for this meta-analysis. In it, we have, this graph is all of the places where we have soil carbon persistence data, so 14C data for different fractions, and then the abundance, so the amount of carbon in the different fractions. So overall, we had 296 profiles, 120 sites, and 51 studies represented in this analysis. Though I will say we are missing data from large areas. So Australia and Africa and Asia, as this database grows, we really hope to increase our coverage across the globe. So I won't go into the modeling, I just want to go into the results. So first, we looked at persistence, and I'll show you how these graphs work. So this is mean annual temperature on the x-axis, and on the y-axis for all of these graphs, if we have persistence, we have this 14C value. So if you remember from the intro, as it gets more negative, we get these older values of -1000, and then in the newer values tend to be positive from the bomb peak carbon. And so generally, as we have mean annual temperature increasing, we see that the carbon on average is newer. And so that means it persists for a less amount of time in the soil. And this we found in all fractions and across all depths. So we really looked at how these soil forming factors interacted with both the depth and soil fractions. So overall, persistence decreases with warmer temperatures. This was generally expected because with warmer temperatures, we have faster biological activity and then faster microbial turnover of the carbon. So it won't be staying in the soil as long. We did see a significant influence of both fraction and depth when it came to the effect of temperature on the abundance of carbon. And so we found that the abundance decreased with increasing temperature in the free particulate fraction. So back in the warming experiments, I hypothesized that the free particulate fraction would be the fraction that's responding at all depths to warming. And so the meta-analysis supports this a little bit. So we do see that the free particulate fraction has less carbon as the temperature is greater, but we do not see a significant effect at depth. We only saw this in the top 30 centimeters. The other really interesting thing to point out here is that the occluded light, so that aggregate associated fraction, and the mineral associated fractions, actually their abundance, the amount of carbon in those fractions, increased with increasing mean annual temperature at all depths. And so I think this comes down to, again, as it warms, we have higher biological activity. And so in the occluded aggregate fraction, that could be of soil fauna like earthworms. So if their activity is greater, we might have the formation of more aggregates. In terms of the mineral associations, it is thought that that mineral associated carbon has been processed through microbes first. And so higher microbial activity might lead more of that free particulate carbon to be mineralized. Some of it would be lost to CO2, but some of it would then become mineral associated. And so I thought it was interesting that these depths seem to act differently in response to temperature. We also saw throughout this project a really significant effect of moisture availability. It was just as important as temperature in determining both persistence of carbon and its abundance. So here we show that the persistence of carbon, again, older carbon is this more negative value here on the y-axis. And this is a wetness index, which is the mean annual precipitation divided by the potential evapotranspiration. So it is a metric of a proxy for moisture availability in the soil, the best we could do at this sort of global scale. And as we go through these panels, we have the surface soils down to deep soils. And so persistence increases with increasing moisture availability, and this effect is strongest at depth. Why is that? Well, moisture is really important for the weathering of soil minerals. And so the more weathered soil minerals are up to a point, the more capacity they have to absorb to soil organic matter. And so you also need enough moisture to be able to develop a deeper weathering front within the soils and to develop deeper soils in general. So it seems that this correlation indicates that moisture can be really important for the formation of organomineral associations and then making the carbon last longer and persist longer at depth. And I would argue that this is likely not responding to like an annual moisture availability, but really the cumulative effects of more moisture over a long periods of time. Lastly, for this meta-analysis, I wanted to show the results of land cover. So here we have the percent of carbon that is minerally associated. So like what proportion of carbon is associated with soil minerals by land cover classes. And for this analysis, climate was not nearly as important as land cover class to determining the proportion of soil carbon that was minerally associated among these different soils. So the temperate forest here has the least amount of carbon that is associated with soil minerals. It's the only land cover that had less than 50% of the carbon that was mineral associated. Tropical forests, grasslands, and croplands all had over 75% of the carbon in their soils associated with soil minerals. And so I think this reflects back on the warming experiments where we saw a strong response in forests to warming, and we didn't see a strong response at depth, especially in the tropical forest where we had an andesol with a lot of mineral-associated organic matter. And then my hypothesis for the grassland and cropland warming experiments is they will also not have a strong response due to having more mineral-associated organic matter. So, in conclusion, wrapping up, yes, deep soil carbon is vulnerable to warming-induced losses, but it's not vulnerable everywhere. And so the system really matters. And the strength of the response of deep soils is dependent on the amount of this unprotected free particulate carbon that is found at depth. And it seems that temperate forests are particularly vulnerable because more of the carbon in temperate forests is found in this unprotected fraction. Globally, the response of soil carbon abundance and persistent varied with soil fraction and depth. But again, it seems that the shallow free light fraction that's not protected seems to be most sensitive to temperature-induced losses. And I would just like to highlight that in that meta-analysis, wetness index was really important. It was just as important as soil temperature. And so I think the importance of the wetness index really shows the need for more global change experiments focused on how changing soil moisture might affect soil carbon. And there's been a lot of drought experiments. We haven't seen a lot of experiments with different types of moisture manipulations. And again, it's unclear whether the effect of moisture is one as over long periods of time or whether we'd be able to see its effect on the order of years to decades. And just like to point out that moisture matters for transport of carbon to deep soils, how much carbon inputs there are through plants, reaction rates in general, both biotic and abiotic, and weathering rates. So I do think there needs to be more focus on this going forward. With that, I would like to acknowledge all the many people who have helped. So we've seen experiments across 4 different sites and 2 different continents and a meta-analysis that was global. And there's just been so many people that have helped out with this. So I would like to thank all of them. And now I will take your questions. Thank you.
Okay, so we have 2 questions so far. The first question is from my colleague Frederick Ritz. He is asking about the experiment and particularly does soil respiration also include root respiration, or was root proliferation limited by tranches or walls around? If root were included, is it not normal to have a signature of recency in the respiration? And in any case, should we also consider warming the arid part of the plant to include the possible effect of warming through an increase in below ground sea inputs? I have an opinion on that, but I will let you respond.
Yeah. No, this is a great question. There's many, many parts to it. So first, yes, this is soil respiration. So it is not only microbial, it also includes root respiration. So we did not make an attempt to split them or to partition them. And it is increasingly seen in many systems that we do see this recent carbon in the respiration, but that's not always the case. For instance, in the Arctic, even though we also are including root respiration, we do, and in peatlands, where the carbon is very old, we do see old carbon signature in the respired carbon, despite there being roots. And so whether you see an old carbon signature seems to be system dependent. And yes, it would be diluted by root respiration, but especially at depth, root respiration is probably, I mean, if on average it's about 50% of soil respiration, and at depth it's probably much less. We'll be generous and say 25% at depth. So I don't think it would preclude us seeing a signal of old carbon if there was one. And yes, we should absolutely consider warming the aerial parts of plants to include possible effects of warming through increased inputs. It's really hard to do that in soils. It's cost prohibitive. In forests, it's cost prohibitive, but we can absolutely do that in grasslands. And I do want to show, I'm just going to, sorry to go through, I can go through the slides here. So in Blodgett, we did actually see an increase in root carbon at the surface. So I focused here on this red where we had decreases in the amount of soil carbon at the surface at depth, But at the surface, you can see, especially in the top 10 centimeters, that we saw an increase in soil carbon stocks by up to 75%. And that, to me, absolutely seems like there's been an increase in root inputs, potentially as a result of warming. Even though we're not warming the plants in the forest, we are warming the roots. And so that could increase their exudation rates. It could increase production. But we did not measure it in this case. And that is definitely something that we should be thinking about.
Thank you, Kathleen. The second question is from colleague Ye, Cho Xian Ye. I'm sorry for the pronunciation. And he's asking a question about short-term versus long-term. And he said that that the short-term experiment, which cannot capture the temperature responses of slow carbon pulled in deep, I mean, this is a question of long-term and short-term and how carbon is responding to that.
So we absolutely were able to measure the response in the forested ecosystems. I mean, so much so that we were even able to overcome that issue with signal to noise ratio and measure a difference in stocks over 4.5 years. So I think if deep soil carbons is going to respond, it's not going to respond slowly, like an increase in microbial activity as a result of temperature is something that happens really, really quickly, on the order of minutes to hours to days. And so if there's carbon that can be respired by microbes at depth, then they will be doing so at an increased rate pretty quickly due to warming. If it's an effect of warming on more like weathering processes or something like that, it could take a really long time to see a difference. But if we're expecting it to be driven by the microbes, if it's going to be a response, I don't think it would be a response that would take a long time to measure.
We have another intriguing question from Lucas Lesen. He said that, can you really put in the same category soils sample at 30 centimeter and soil sample at 167 centimeter when talking about deep soils?
So no, I absolutely wouldn't. And they were put into this category for visualization purposes only. This meta-analysis, depth was the most important factor in this model. And so depth was absolutely taken into account. And so this was me trying to distill a complicated meta-analysis into a simpler graphic. And so the best way to show this to you was by putting all of these together. So that's why it was in this graph, but we absolutely know they're not the same. Though that being said, if it is about sort of mineralogy and weathering front, it depending on the system, 30 centimeters, like the one I had in New Hampshire, might be sort of where you have a lot of those minerals being accumulated. And in another system, like a tropical system, maybe that's being seen much deeper because there's much deeper weathering in that soil. It's a much deeper soil. But yes, good point.
Yeah, we have another interesting question from Mohammed Salim. He's asked, what would be the overall impact of bulk density and say-in ratios of varying soil depth?
Not sure I quite understand that question. So the bulk density increases with soil depth. And so I think the overall impact of that would be potentially more carbon being inaccessible to microbes. If you have a higher bulk density, perhaps there's more micropores, and so more of the carbon at depth might be inaccessible to microbes as a result of that. C to N ratios, those are really highly variable across systems with soil depth. And generally we would see lower C to N ratios at depth because the carbon is more microbially processed. In the forest, however, particularly in Blodgett, the C to N ratio is actually, it decreases at first with depth like you would expect. And then around 60 or 70 centimeters, it actually starts to increase with depth again. Again, showing that Blodgett did have a lot of this carbon that was not very microbially processed, and that was really like ripe for responding to warming in this case. Mm-hmm. And again, there's not many experiments like this, so we can't say to what extent that the Blodgett forest response was typical.
Thank you, Kathleen. We have another question from Rose Abramohom. And then the question is for the Blodgett figure showing more losses in the particulate fraction at depth, it seems like there was perhaps the opposite effect at the surface. Am I interpreting that figure correctly? Either way, how do you interpret the surface response at Blodgett? Do you think that mineral associated organic matter is just not temperature sensitive?
So here, I think Rose is referring to this graph where we showed that there was this decrease, so less carbon in the particulate organic carbon at depth. And if you look at the surface, you see that the warming actually has more of this particulate carbon. Again, this goes with this graph, which actually shows an increase in the carbon stock at the surface as well. So whatever that increase in carbon stock is, it seems to be driven by the particulate organic matter. And again, I mentioned it could be roots that's driving this, but it also could be, it could be actually a response of the microbes to the warming, and perhaps they were actually less efficient. And so we might have a buildup of organic matter in litter layer, potentially that increases the amount of particulate with warming. So again, we don't really know what's happening in terms of causing that increase. Do I think that MAUM is just not temperature sensitive? I don't want to say that, no. I think it's a lot less temperature sensitive. It seems, the data I have seems to show that its temperature sensitivity might actually be towards an increase in MAUM, at least in the amount of carbon associated with MAUM, as you get warmer temperatures, because you have increased microbial activity. But I don't want to come out and say it, but that's what I'm thinking. I want more experiments, but that's what I'm leaning towards, that it is not as temperature sensitive.
We have another question from Edwu Atolio. I'm sorry for pronunciation. To what degree does increased climate warming affect deep soil temperature? Would the outcome and change in soil organic fractions under agriculture versus natural ecosystem differ? We will see.
Yes, that is, we are hoping really to see that. I think that's the most exciting thing about this collaboration, is that we'll be able to look at these processes in an agricultural soil, in a soil that, you know, has a really different, it's really different in terms of the amount of particulate versus mineral associated carbon that it has. It's very different in terms of the amount of disturbance that it's already experienced. And so, yeah, we'll wait and see. And just wanted to bring back to this graphic here that the models, and there's also one paper I can't remember off the top of my head, that actually shows data that does show that the deep soil is just as responsive to warming as a surface soil. So deep soils are warming. In general, under steady state conditions, the mean annual temperature at any depth in the soil is equal to the mean annual temperature of the atmosphere. So while there might be a short lag, by short, I mean like a year lag, as the air warms, to see that in the deep soil, you'll absolutely see that effect in the deep soil.
Okay, we have another question. I think they're from Marie Arnaud. She's asking, have you investigated tropical wetlands, tropical peatland mangroves, since soil water content seems important for soil carbon persistence? If not, how do you think deep soil versus shallow one, carbon will respond to temperature in this ecosystem? I think it's coming soon also.
Yeah, I think that's definitely something that it should be being investigated. I think for that, it's so hard to tell. So in those systems, if they stay wet, what will the effective temperature be? Oof. I'm guessing they will still be responsive to warming, but not to the same degree, since the decomposition is really limited by oxygen in those systems. Though that being said, I would encourage you to look at the spruce experiment, which is being run by Paul Hansen at the department at Oak Ridge National Lab in the US. So this is an experiment where they've actually warmed an entire ecosystem, an entire spruce bog, and they're looking at the effect of warming on everything in that spruce bog. And so perhaps, even though it's a much colder system overall, it is an inundated system, and so you might get answers there.
It's another long question from Yang Su about the relationship between soil temperature and air temperature in your study. Do you consider the impact from soil cover and different type of soil covers, such as fallen leaves or residues? Question mark. The temperature differ from soil covers will also affect the soil organic matter content in different soil depth, since there is more carbon input, low evaporation, and probably lower decomposition rate due to a lower temperature from soil cover in the hot days.
So with all these analyses, I also looked at the actual soil temperature at the time that I took the measurement. So those differences would be taken into account in terms of how soil cover affects it. In terms from an experimental point of view, the soil cover and the amount of water in the soil matters a lot for how even the heating that your experiment will experience is. So generally, more deeper litter layer and more wet soils will have a much better, more even heating treatment with these deep soil heating rods than a drier soil with less cover would. So that's sort of like the effect of soil cover as sort of an experimental artifact. And again, yeah, carbon inputs, that definitely matters. And that is one of the issues, is that when we get to these forested systems, in terms of leaf litter, it's not affected, its production is not affected by warming at all. But once it gets to the soil, its decomposition will probably be affected. And then I see.
Yeah, your mic is turned off, Abad. It's not, I don't think it's a problem. You see the last question,
okay,
I guess.
It's okay, I capture it, yeah. So we have a question about from Dansu. Do the different soil fraction methods in the lab give very similar result from the proportion of different pools?
So if anyone knows a good study that compares that, please let me know. One issue we've had with soil fractionation is that and this was an issue with our meta-analysis as well. The density fractionation is very popular with people in forest soils, and then a size fractionation method has been a lot more popular in grassland and cropland soils. And so there is a bit of bias in terms of the ecosystem and the field that people are coming from. And I think we need to have more comparative studies that are looking at, the size fractionation versus density fractionation, especially when it comes to radiocarbon ages. A really good way to tell if you're getting distinct fractions through these methods is to look at whether you have distinct radiocarbon ages of your fractions after you've done the fractionation.
Thank you, Caitlin.